actually remember as a starting PhD student, master student in fact, and these first lunches when the more senior students were really discussing the human frontiers. Will I get the human frontiers? And I didn't understand what it is. And of course they were discussing the postdoctoral fellowship and the passion they spoke about it, how much they wanted it, and of course the name, which human frontiers, already gives this sound of really something going to the next frontier, the next stage, and this very ambitious thing. And I, of course, this thing stayed with me for many years, and I realized, of course, as you've seen, how important this program is, and I was very fortunate after all to get a postdoctoral fellowship and a grant later on. It's really, to be here now with the Nakasone Award, it really brings out these feelings and how grateful I am for the great efforts that Human Frontiers has done for science, for different fields, for postdocs, really all over the world. And again, I want to congratulate Human Frontiers on the great projects, and I look forward for their continued projects for the years to come. Of course, I want to thank, I wouldn't be here without mentorship of my PhD advisor, Professor Ofer Mandelbaum from the Hebrew University, where I started in immunology, and later on with Professor Rudy Yanich for my postdoctoral training, iPS cells. I've learned a lot of them much more beyond science, and I'm forever grateful for their support. And of course, I'm very grateful that I've been very fortunate that my lab was set up thanks to generous and wonderful couple, Ilana and Pascal Mantou from Israel and France, who helped me establish the lab and continue to support our research, which I will try to show you. It's very technically demanding and resource demanding. And of course, there are other anonymous philanthropists that continue to support this science. I'm thankful for my institute and board for a lot of support and infrastructural support, which is very critically needed. And of course, my family, my parents, my partner, my daughter, and my friends at the Institute and outside the Institute that really allow us to go through difficult time and good times. With that, of course, and then it comes to the people who are really doing the work. And I've been very fortunate to have a lot of very talented master students and PhD students, again, from all over the world, whether it's from Israel, from Palestine, from Austria, even Luxembourg, and of course, collaborators in Israel, outside Israel, that allow us to do these projects, and of course, funding agencies, including Human Frontiers, to really collaborate with scientists from other parts of the world and discover new technologies and new bonds which have lasted long after the grant has ended. And with that, I'll start telling you about this journey that we started, our work on stem cells and how it's kind of got into this embryo-like modeling research and why we are doing this. And as you know, one of the challenges in modern society is really that as we age, we are facing more diseases. There's great need for organs and organ transplantation and replacements of tissues. And this is a major problem because it's very hard to find donors. And even if you find donors, there are issues of rejections and so forth. And I really like this painting from Cranach from the 16th century. It's called The Fountain of Youth, where you can describe this rejuvenation cycle where you have people who are a bit older and perhaps sick and come and bathe in the water of this fountain and go out on the other side very young and cheerful and naked and perhaps going on to celebrate further on. And there are even more modern takes of this, of course, which is by the New York Times. And I like this image because it talks not only about disease and illness, but also needs about having a higher quality of life as we age. People want to play tennis when they are older and hike. I always say 80 is the new 70 and 70 is the new 60. And this is something that is being required in modern society. And our view on this cycle of rejuvenation is what happens in development, where as you know, we all start from a single cell, which is the fertilized egg that becomes the zygote, that grows and becomes, at the 64th cell stage, the blastocyst with the inner cell mass. And this inner cell mass, these stem cells that are called pluripotent, they are in a way considered the climax of developmental potency. And then the embryo implants in all mammalians and starts to grow. Still, pluripotent cells are still found in this stage. And later on, the embryo gastrulates, which is making the progenitors of organs, and then organ formation. And already at this stage, some cells called primordial germ cells, PGCs, which are later going to become the sperm and oocytes in puberty. But these cells are already set aside very early in development. And of course, in the next generation, these cells continue to transfer the genetic material. And this is really the rejuvenation cycles that happen in mammalian life. In my lab, we're interested in different aspects of studying different things. A lot of dramatic things happen very rapidly during these rapid transitions. For example, you have different states of pluripotency before implantation, after implantation. You have cells which are called totipotent in the earlier stages that make the placenta. What dictates the placental fate versus the inner cell mass fate? What is specifying primordial germ cells? How similar are they to pluripotent cells? And of course, we are very interested in this in across evolution, human versus mouse, and how these things function at the molecular level and both at the cellular level too. And when we start to talk about pluripotency, we must remind ourselves that pluripotent cells, unlike other types of stem cells in our body, it's actually a transient state. So pluripotent cells exist for about 1 to 2 days during our development. We do not have them as adults. This is not like skin stem cells or intestinal stem cells that exist and self-renew. So this is a transient configuration, and there is no really self-renewal in vivo. These cells emerge and then resolve. But 40 years ago, Evans and others were able to grow these cells in the dish, and this is a synthetic artifact. It's a very useful artifact that we can grow them in the dish, and they could do it by giving them the right cytokines. If you give them these cytokines, you basically prevent them from moving forward. Typically in the mouse, the signaling pathways involved are lift stimulation, Wnt pathway stimulation, and inhibition of ERK. Typically, these conditions are called 2I-LIF as an acronym, and I will talk about it. And this is how we keep stem cells. And of course, later on, there was this new kid on the block, which is iPS cells, a discovery by Yamanaka from Japan, which he showed that pluripotency can be reawakened in fibroblasts when you give defined factors. And what Yamanaka really showed that you can take any adult cell type and express these 4 transcription factors ectopically. And in a rare fraction of the cells, you'll start getting these colonies. You can see here, very rare, about less than 0.1%. And if you carry and grow these cells further, you can get these induced pluripotent stem cells. And they are, in the mouse, what you see, they are really indistinguishable from embryonic stem cells. You see here, each one is like a colony. It has about 200 cells. And these naive cells that are early are like kind of have this domed, shiny looking cells growing in there. And of course, the great promise of the stem cells, and this is something I did when I was starting as a postdoc, and this is kind of perhaps the dream scenario, is how can we use these iPS cells to treat diseases? And we took these mice that have genetic diseases. It's a humanized mouse model for sickle cell anemia. So have a mutation, the human beta globin mutation, which it's actually 10% of African Americans are carrier for this mutation. And it's a very bad disease because under stress, the blood cells become sickled and block blood vessels and so forth. And the idea that we did is to take skin cells from these mice, grow them, express the 4 factors transiently, and make genetically identical iPS cells. And of course, one thing iPS cell technology gives us, ES cells and iPS cells are very amenable to gene targeting, so we can correct the mutation. We don't want to transplant sick cells, just correct a mutation. And then you have genetically corrected, basically, iPS cells that are identical. And then one principle with pluripotent cells is that after we go backwards, we have to differentiate them into different cells before we transplant them. So this whole concept of pluripotent cells, let's say we put them in the lung and they will learn from their neighbors, it doesn't work actually. If you do that, they will make very benign tumors of different tissues. So we really need, and which is a major challenge, how do you differentiate the cells properly and purely? So we use some kind of a protocol to make something like a bone marrow cells, but not exactly. And then we can transplant these cells, and we showed that 80% of the mice were cured from sickle cell anemia, which was an exciting demonstration. But of course, since then, the challenge, how do we make this a safe and realistic reality for humans, which is a really complex issue. So in my lab, when we wanted to approach this, and perhaps because as we were very after basic research, not necessarily applied, we looked at different type of transitions, which one of them was, how do you take a differentiated cell and convert it all the way back to this naive pluripotency, which is like the inner cell mass. So naive really talks about this maximal level of pluripotency. And another aspect is, how do we convert different type of pluripotency, less naive or what we call primed, and also then how do cells exit pluripotency, which is actually overt differentiation, or how does pluripotency resolve and become something differentiation. And the idea was to really understand the molecular machineries, the principles, the boundaries systematically, to understand them and compare them, what happens in the dish in vitro versus in vivo, what does happen in human versus mouse, of course, maybe other species that happen there. So I will talk just a bit on the first aspects, and actually this is something that was funded by Human Frontiers, which I was very excited about. We wanted to ask, what are the molecular machineries involved into converting a skin cell back to an iPS cells? And there are questions about the how and the why and the what. What enzymes are involved to remove all epigenetic marks and cells become iPS cells? How similar are these in vitro reprogramming to in vivo reprogramming events that I showed you before? Why do most cells fail to convert, and change that? And what is the molecular trajectory of successful reprogramming? These were all unknown questions with unknown answers. So just to give you a couple of examples, we were able to uncover several molecular important regulators of these processes. In this case, an enzyme called UTX, which removes H3K27 trimethyl. So this is a repressive epigenetic mark. And we find that if you knock out these enzymes, you cannot make iPS cells, and actually you cannot make germ cells. So it's important for the reprogramming of both these cells, in vitro and in vivo. And UTX is brought by the Yamanaka factors to these repressive marks on the pluripotency gene. So it is really important to de-repress pluripotency genes and be successful to make iPS cells. So it's not a rate-limiting factor, but it's an essential enzyme that kind of cleanses these repressive marks to reactivate pluripotency. In the different players we were looking at, we were asking, what is the rate limiting? Why do most cells fail to reprogram? And we already knew that we could activate a lot of factors, but this is not enough. The efficiency will be about 1%, 10%. But the question was, can we really make the reprogramming become almost deterministic, meaning nearly all the cells will just go back to become iPS cells? And we thought something differently. Perhaps instead of adding activators, we should just remove repressors. And what we found by a screen for repressors is that we inhibit one repressor called MBD3, which is very poorly studied, and we need to inhibit it in a hypomorph level. I wouldn't get into the details, but the nice thing about this is that we can really reach up to 99% reprogramming efficiencies when we do this. And you can see this video versus in the wild type and the reprogramming of the MBD3 low, and the fibroblasts are red and the iPS cells are yellow, and you can see really on the left how nearly all the red cells become yellow at the same time. So it's very synchronous, efficient reprogramming compared to the wild type cells. We later on started to dig deeper, understand what is happening, and what we found that reprogramming mechanisms in a way acts what we call the gas and brakes models, meaning when you bring these Yamanaka factors to make iPS cells, they recruit activators like UTX that I showed you to turn on pluripotency genes, but actually they also bring repressors that like the MBD3 I mentioned to you to the same genes. So this is what is happening is equivalent to trying to drive a car while you step on the gas and the brakes at the same time. When you do that, the car drives sometimes, but it's jumpy, so you will get some IPS cells. But if you take down the handbrakes and step the gas, then you will get very efficient, smooth, efficient reprogramming. And we dug a bit deeper, and this complex is not very known. It has many different conformations, and we can really find other ways we can knock out another component of the complex completely, so we don't have to work with hypomorphs. And this is called GATA-D2A, and if we remove it, you can see here really 96% efficiency within 8 days, just like as I showed you before. So this was really exciting because not only it uncovers about concept of blocking how do you make naive pluripotency, but it also started making ways to ask what happens during successful reprogramming. So typically when you look at iPS cell reprogramming, it is a process which is very inefficient. So if I want to map the chromatin changes and 99% of the cells fail, I don't want to look at those. And I can't do much sorting because when I'm at day 3 or 4, I cannot a priori predict which cells are going to make it. But now with this system, I have a system where it gives me 99% successful rate of reprogramming without having to do any selection. And we asked, what does the chromatin and changes look like? And this was, again, the project funded by HFSP with Will Greenleaf, and we were able really to go and map transcription, chromatin, DNA methylation of successful reprogramming every 24 hours and learn a lot of mechanisms of really how the chromatin changes and what precedes what. About, for example, the transcription factor binding and the changes of the chromatin marks, what is the role of MYC, which is found to be very different from the other factors, and so on. So this was a big part of our research, and we, sorry, excuse me. And we learned about, and we still have a lot more to learn about how naive iPS cells are formed and reprogrammed. But at the same time, we were also studying a smaller transition, which is how do stem cells get different configuration? And I mentioned to you, typically embryonic stem cells represent the blastocyst, which is the naive pluripotent state. But pluripotency also in vivo, this is the mouse, after implantation, you still have OCT4 positive cells that are about to differentiate. They haven't differentiated this. And this is called prime pluripotency now. And different groups at the time in 2007, we were able to grow them, and they require different cytokines, FGF activin, and they can be stabilized and grown in vitro. And over the years, my group has spent a lot of time to really dissect the differences between these states, which are both pluripotent, but there are differences. Some genes, like ESRRB, is expressed only in the naive state. They have different signaling requirements. They have different metabolism. X chromosome inactivation, for example, already occurred in the female cells in the prime because they're about to differentiate. DNA methylation already is increased. And usually the naive cells, the early ones, are the ones that are really good at colonizing the blastocysts, and that's why they are used to make mouse chimeras and germline transmission, versus the prime cells are more advanced and have limited developmental potential. What we've learned over the years that what dictates the states of naive or prime, it's about the media that we are used. So you have two different cell types. So you can have ES cells, which is embryo-derived, and you can have iPS cells. But there's also a cell state. So an iPS cell can be a naive or a primed state. And this is dictated, theoretically, this groove is by the different cytokines we give. And we stabilize the cells in either one state or the others. And we can just change the media. We can just take naive cells, they become primed, or prime cells can be reprogrammed back to naive just by changing the signaling between the two, which was an important principle. But this has started to raise questions. What about human stem cells? For years, human stem cells were very cumbersome to work with compared to the mouse cells. And the mouse cells, as I showed you, they have these domed-like morphology. The human cells are very flat. They're hard to grow as single cells. Also, they are very similar to the mouse primed cells, and the same, they're epithelial and they are flat and so forth. And for years, scientists in the field accepted the human stem cells for what they are as they are, because the notion was, well, it must be evolutionary differences because they were derived from the blastocyst, so they must represent the blastocyst. But as I showed you earlier, that is not necessarily the case. What you start with is not necessarily what you end up with. There could be a theoretical state or situation where you take a human blastocyst, and because you haven't made these conditions correctly, the cell just differentiated and assumed a later developmental state. Therefore, there was a question, was there a human state which we have missed all along, which is what we call a human naive state? And we know that the mouse naive cells conditions are not good enough to isolate human naive cells, but still there was a question, maybe we need more factors, maybe we need different factors. And maybe this is a reason for the limited functionality of human stem cells. So we started with this project. It was Ohad Gafni 2013, and our question was, can we make an alternative pluripotent state in humans? And in our opinion, when we look at these naive and primed neighborhoods, the major divider between them is how they respond to blocking of ERK signaling. Naive cells tolerate blocking FGF-ERK signaling, in fact, prosper in this state, versus primed cells differentiate and deteriorate under these conditions. Therefore, our goal was, can we make human ES cells that can tolerate blocking of MYC-ERK signaling? And indeed, we found out that we can take these mouse to ILIF conditions, and in the human, we needed to add more factors to inhibit PKC pathway, P38 pathway, provide activin, which was surprising at the time. Now it has become consensusly accepted. And the bottom line is we can make alternative cells in humans. You can see them here dome-shaped that are very different from the conventional human cells, and they have different molecular properties and so forth. But since then, they were started asking, well, we knew that we made this first jump to the naive neighborhood, but we asked how much more naive can we make? And since then, we've learned with single-cell technologies on human embryos, we knew a lot of more properties about human inner cell mass. And we knew that our cells were not already all the way up. And we wanted to make these enhanced conditions. But there's a problem when we work with human stem cells. How do we define functionally a human naive cell? In mouse, we can inject it in a blastocyst and put it inside a uterus. And if it makes a mouse, that is naive enough. Obviously, we cannot and do not want to do this with human cells. So we were looking, is there something functional properties that we can think of for making human naive cells, or fully naive cells? And we stumbled on this from the work of Abid Mansour and Shai Geula. Abid is now a professor at the Hebrew University, who wanted to look at, are there specific regulators in the mouse of primed pluripotency? We knew that there are factors that are important only for naive. We knew the factors that are important for both primed and naive. But there was no evidence for something that is only important for the primed stem cells. So they've made these OCT4-GFP, which is a reporter of stem cells, naive and primed. And you can knock down the different genes and see if the OCT4-GFP goes down, it means it differentiated. And we focused on a bunch of epigenetic regulators. And indeed, as you can see here highlighted in gray, there were multiple genes. When we knocked them down, only the primed pluripotent cells deteriorated. But if you look carefully, there's a common denominator to all these genes, that they were all some kind of a repressor. So DNMT1 is a DNA methylation transferase, which is a repressor. Dicer and DGCR8 make microRNAs, which is a repressor. EED and Polycomb make K27 trimethyl, which are a repressor. And in this case, even METTL3, which we focused on, it's an RNA methyltransferase that leads to degradation of RNA, so it's a repressor. So there seems to be perhaps a concept, perhaps a little bit philosophical, first, that naive pluripotent cells are very minimal in their requirement for these fancy gene regulatory mechanisms. Meaning, if they have microRNAs, but if we remove them, the pluripotency doesn't collapse. Again, if we remove DNA methylation, they don't collapse. And perhaps in a way, They are similar. If we ever had a unicellular ancestor, perhaps yeast, they're similar to that. So as you know, yeast cells do not have DNA methylation, do not have microRNAs, do not have RNA methylation, only in meiosis. Human mouse ES cells can grow as haploid cells, which is very unique for cells, and this is similar to yeast. So it's really start to think that this is a very basal state, and it's very unique, and it's unlike any other cell type, whether it's even primed cells or somatic cells in development. But more practically, this gave us a screening property, and now our question was, can we make human pluripotent cells that tolerate removal of these enzymes? So it's not only about growing in ERK inhibition, can we remove these enzymes? So in this work, we made engineered cell lines, and you can look at MEQTL3 enzyme. We knock out the endogenous allele, and we have a TET-off exogenous allele. So the expression is given only from the transgene, and when we give doxycycline, this enzyme is turned off. And now we can really screen. Now we just give dox and see if the cells tolerate this loss or not. And really it turned out, and this is the work of Jonathan Byron and Sergei Yaqub, that we were very close in our first generation. And basically we, instead of Wnt stimulation as we do in mouse, we need to do Wnt inhibition. And if you do that, you have naive pluripotent cells that are really have a higher level of naivety. They can tolerate, you can knock out all these genes, and they will be fine in their pluripotent state. But bottom line, I would say from all these studies, from us and other groups, we've closed the gap between the different pluripotency flavors we can isolate in the dish, in the mouse, and the human, and we can really start doing comparative studies in development. But all of this research, and I face the existential question, who cares? Obviously, we can do a lot of good things with conventional human iPS cells and stem cells. But what I'm showing you, this is evolutionary conserved, and likely there is a benefit of this. And there is a benefit if you look one example, which is the work from Michinori Saito in Japan, who was the first one to be able to really make authentic PGCs from mouse ES cells. And his secret for success, he had to start with naive cells that were ERK inhibited. He will then, like in development, give them priming for 48 hours with FGF activin, and then he will pulse them with a bunch of cytokines, mainly BMP4, and he can get these Stella positive, BLIMP1 positive, double positive PGCs, which if you transplant into a testis or an ovary, will go on to give rise to a sperm and egg. When this was published, many people tried to do this with human stem cells, and they failed. And they thought, well, maybe different timing, maybe different BMPs. But at the time when we looked at this, well, everybody, the human stem cells were using primed cells. What if we use just naive cells? And really, we turned up with Azim Sorani, and this perhaps a rare event where experiment works from the first time. When we started this in humans from naive cells, whether it's the old version or the new version, we can make human PGCs, authentic ones, in the Petri dish. So the protocol is the same between mouse and humans. And what this is showing us, at least, that the starting state of the cells, it has a critical influence on the differentiation success or failure in this assay. And there are very big questions, which now bring us to topic 3, which is differentiation. If you look in this PGC paradigm, there's a question, why is starting with naive cells critical? Why do we need to go through this priming to differentiate? And also, if you do priming for more than 2 days, PGC competence goes down to 0. So I'm talking about, in a way, theoretically, windows of competence that open up at the right time if the right cytokine giving at the right amplitude. And this is a bit of a scary scenario for us because this can be happening beyond the PGC paradigm. If we looked at human iPS differentiation, even in the mouse, we have a really hard time. Most cell types, we are not able to make them from iPS cells. Even HSCs in the mouse, which we know how to transplant, nobody has been able to make HSCs in the mouse after 40 years of research. And maybe if you're taking the HSC paradigm, and this is actually, you have maybe 10 stops like this where the right thing needs to happen at the right time to succeed. And therefore, we actually started to ask, we want to dissect this like an enzymatic reaction. And if we want to study enzymatic reaction, as you know, we need to capture everything in the dish. And we were stuck in our research because we can go back and forth between naive and prime stem cells easily, but I don't have a readout for my differentiation. Because if I want to study this sequence, and if I want to introduce perturbations for this sequence, I need the naive cells and the prime cells become gastrulating embryo-like entities, which is now what we call stem cell-derived embryo developmental models, or SEMs. But in order to do this, which is quite ambitious, because I'm actually trying to make developmental model and grow it outside the uterus in a dish in the stem cells, but I actually can't do that even with natural mouse embryos, because we cannot grow mouse post-implantation embryos to become gastrulating embryos. So how am I going to do this with just stem cell derived? And therefore, we thought maybe we should try and solve this problem. And if we are successful, we can backpedal and try and address this first question. There's another side of the coin for this kind of thinking strategy, which, as I told you, typically differentiation protocol to make, let's say, HSCs, they are directed differentiation. So we, the scientists, decide when and which cytokine to give and trying to make a certain cell type. But you might argue, well, Yaqub, maybe your iPS cells are the problem and they're not good enough. But that is not really the case, because if we take mouse ES cells and iPS cells, inject them into an embryo, put it inside the uterus in the mouse, make a chimera, the bone marrow cells are just perfect. So the problem is not the cells, the problem is us, the scientists. We are not able to recapitulate properly yet the sequence of events to make these stem cells differentiate. Therefore, we faced a new challenge, which was not something we planned for, and we had a kind of a multi-step strategy, which is the idea, let's try to grow mouse embryo post-implantation for very long times. And if we are able to make these conditions, then let's try to put mouse stem cells under these conditions and see if they can make a developmental model of embryogenesis. And of course, eventually, we are very much interested with human SEMs that are formed in this way. And of course, this is very important for not only to study stem cell differentiation, but to study basis processes of gastrulation. So gastrulation is the process where really the stem cell ball basically starts laying out the footprint to make organs afterwards. And Lewis Wolpert, a very famous developmental scientist, had this famous quote where he said, the most important day in our life is not when we are born or we die or we marry, but it's actually when we gastrulate. Because if we gastrulate correctly, we don't have developmental defects. And really, a lot of developmental defects, although they are uncovered much later on, they happen during gastrulation. So this brings, of course, in mammalians to what is called the uterine barrier, where, as you know, we can grow mouse or human embryos until the blastocyst stage very well inside the dish. That's why IVF treatments exist. But once you reach this stage, typically you have to either put this inside the uterus, and this in IVF, or you freeze the embryo. But what we are interested in is these stages. Once the embryo implants, then quickly we have this, what I call the drama, where the embryo starts making its organs very quickly within a number of days. So basically, by day 12, nearly all organs in the mouse embryo are formed, and the rest until day 20 is just basically growth of the embryo. And this is very hard to study, as you can imagine, because first, the embryo is very small. Uterus is not transparent. We cannot control the environment in which this is happening, and this is limiting our research ability. Another aspect of this uterine barrier, which is, of course, in the mouse, if we take out a post-implantation mouse embryo at day 6.5 or 7.5, it's game over. We cannot put it back inside the uterus. It doesn't implant. Then, of course, if you come to talk about humans in these stages, it's really nearly impossible to do any type of research because there are justified ethical and technical limitations on this. Typically, all organs form within the first 5 to 6 weeks of human pregnancy. But usually, women don't know they're pregnant in this time. And even if there is knowledge and there is somehow ethical approval to get some sample of fetal material, This is very rare specimens, which are very useful to make atlases. That's why we have these Carnegie collection and so forth. But this is not a viable way to look at human embryogenesis. So this uterine barrier really has multi-level interpretations. So when we go back, there's a bit, perhaps even a philosophical question here, which is in general in mammalians, does the uterus dictate the shape of the embryo, or it actually doesn't, and we can perhaps be able to capture gastrulation in the mouse outside the uterus, or even capture gastrulation or organogenesis outside the uterus properly, which is something we wanted also to answer. So we're not the first one to try to grow mouse embryos, and you can see papers going back to even 1934 from Dorothea Rodnick. There's been really great work from Dennis New and from Patrick Tam in Australia, but they were able to grow mouse embryos for only about 1 day, and most of the growth was abnormal. But what we were after is trying to grow very long periods of times without delay in mouse embryogenesis. And this is work that was led by Alejandro Aguilera Castrejón, who just actually recently started his lab in Genili HHMI in the USA. And his idea, together with Ghada and Ranan, is to try to do this in a very controlled way and try to build an incubator, try to have a controller system where we control different parameters, which we didn't know at the time, have a gas mixing box where things get mixed, pressurized, and measured properly, and then circulated in this system. And of course, rounds of engineering and engineers that took over 7 years led us to this not very pretty thing, which we built in our own garage, basically. But it works. And you can see here, this is the incubator. You have the wheel here where the mouse embryos grow inside this bottle. You have the electronic gas regulation module, and you have where the gas mixing box, and you have this humidifier and pressure controller. And we've published now a Jove. You can really look at the different parameters. We host in our labs for no profit. Anybody who wants to learn the systems, anybody in academia can get the system for no profit. And what we actually can do is we've learned during protocol optimization is that there are important parameters. So for example, you need to control O2 and have hypoxia in the earlier stages of the protocol. We've also learned that atmospheric pressure is important. It is actually mimicking what happens inside the uterus, which has a little bit of a high atmospheric pressure. One Euro solution, but it's critical, is to have a diaper or black cloth, because embryos do suffer from phototoxicity if we leave them for very prolonged periods of time in light. We also need to put them in a glass bottle that it's rolling, because embryos are very adhesive, and if there's adhesion, that immediately gets distortion. We also had to develop the media in which they grow, because they are fed by diffusion. And it's a mix of rat serum and human serum that are freshly isolated to avoid hemolysis, parameters like no phenol red, high glucose, HEPAS buffering. And when you do this, you can see the following: really success of over 85% to start from day 7.5. And you can see in the mouse, in 1 day later, you get the yolk sac expansion, you start having the first heartbeat. 2 days later, you can see embryo turning the head, the feet on the other side. You can see here, day 3, the heart beating. This is blood. This is the blood of the embryo. So there is no maternal blood in this system. And they grow until day 11, which is about 0.8%. And they stop growing because they're too big to rely on diffusion. And I just show you here really that this is very efficient. And so the control here is really the optimized protocol. And just now to show you what happens if you don't have correct atmospheric pressure. You have abnormal embryogenesis. What if you don't have enough glucose, or you have constant O2? You have abnormal developmental embryo models. What if you use the wrong serum? Again, very bad embryo development. So it's a very efficient protocol without delays, but it needs to be done under the right parameters. Then this was exciting, but not enough, because we can start from day 7.5, which is late gastrulation. But we wanted to make the principle that we can do this from pre-gastrulation, which is day 5.5 and 6.5. And here what you can see is that if we just put them in the roller culture, they actually get abnormal embryogenesis in the mouse, which is not good. But what we did is actually we used the same media we developed called EUCM, and used these static special plates called IBD plates And then we can get the gastrulation happening in over 90% efficiency. And you can see here under imaging really how the primitive streaking formed and the somite is formed. This really, you can get this nucleation of embryogenesis happening in this model. Next, we just combined for the first time a static protocol with a dynamic protocol, and we can really capture from 5.5 to day 11, 6 days of development outside the uterus. And again, it's not about the numbers, but about the critical stages, because this captures both gastrulation and late organogenesis continuously outside the uterus. Of course, this is published, that we've done a lot of assays to really show that they have a really very normal organogenesis in the mouse that is developing under these systems. So we were excited about this, and this is really teaching us that really, at least in the mouse, the uterus is not needed to dictate the morphogenesis of the embryo. And there's really a lot of self-organization. This is inherently coded within the embryo to develop its feature, but rather that the uterus is a very good way for providing metabolism for the embryo. Since then, we've been very excited. We've made now, we've teamed up with Oculabs so that they can make the same incubator more stable and available. Again, I don't make any profit, for labs worldwide that they can obtain this. And we can grow now mouse embryos up to day 13 and a half in development, which is really completion of organogenesis. We've also now optimized protocol. We can start this from day 0 and reach day 9 and a half in development. So there's advantages for those who are working on earlier stages of mouse development. But there was a question, is this only specific for mice? And does this work for other mammalians? I want to say rats are very exceptional. They're the outliers because the epiblast has this X-cylinder shape you can see here. But all other mammalians, the epiblast remains flat. And this is called a bilaminar disc. So we wanted to see, can we do this from bilaminar disc mammalian, in this case, the rabbit? And again, there are different things between the rabbit and the mouse that we need to adapt. But this is unpublished, but we are able to do this. We can go in the rabbit, which is a bilaminar disc. We can see here in static condition the gastrulation. Then we can move to the roller culture and reach also day 12 in development. So bottom line, this can be extended to other models for research, and this is not a mouse-specific phenomenon. As a side note, I want to talk about just a bit techniques, because if we want to work on mouse development, as you know, mouse has been a very useful tool to study development, but we just don't want to just grow them. We want to be able to manipulate them and look at different features. So one thing we can do is we can inject lentivirus that is concentrated in the proamniotic cavity at 6.5. In this case, it's innocent GFP. And you can see the embryo just gets labeled entirely without any delay. So they really tolerate this process. And we can now put CRISPR and knock out 2 or 3 genes and follow the same embryo every day, how it is affected. As I mentioned to you before, this is something we couldn't even do in utero, because in utero, once we take the mouse out, we cannot grow it more. But in this system, we can do this. Another aspect is really combining this with development, where at every stage on this roller culture, we can take out the embryo, in this case at day 9, and use this media, the EUCM media. We mount it on the confocal and do in toto confocal imaging on one organ. So this is the neural tube, and you can see how it closes by this zipper-like movement that is known. Another principle is really we finally have a system where we can control the environment of the embryo and watch how drugs or metabolites affect embryogenesis. And this is very important because, as you know, FDA now, any drug cannot be tested on pregnant women at all. They just don't want to take the risk. And there is no way to assess these drugs many times. And we showed now here as a principle in mouse embryos, we can take valproic acid and just add it, and within 24 hours, we can see the known, these are open neural tube defects that are known to influence the embryogenesis. So this could be really a promising tool in the future for looking at teratogenicity. But now I want to, let me skip this. I want to now move to the second goal, which was really then, can we make embryo models from stem cells? And here I want to define what is an embryo developmental model that we want to make. And in our our opinion, what is really important is to make a structural entity. Because an embryo-like entity has different compartments, and they have to be juxtaposed one to each another in a correct form. And they also then have to have a structural developmental dynamism, meaning that it's not only about making a structure and you just look at it, but you want to see it evolve into something else. And really, there's been great work in trying to make some embryo models before, but there was a problem. You could never evaluate them because, as I told you, even for a natural embryo that is post-implantation, you cannot put it in a uterus. It just doesn't implant. But now we have a system, a device that supports natural mouse embryos, and we can ask what happens if we take mouse stem cells and which mouse stem cells, and will they make an embryo or developmental model? And in our opinion, what is bona fide definition on an embryo-like entity, that it goes through gastrulation and completes it. And this is what we were after. And if we look at the mouse embryo, it has three compartments. It has the placenta, it has the yolk sac, and it has the embryo proper. And we got a clue from working on human naive pluripotent cells is that something we noticed that human naive pluripotent cells can very easily become trophoblast stem cells, which are the progenitors of placenta, or primitive endoderm cells, which make the yolk sac. And I mean easy relative to mouse. There is no need for transgene overexpression. So this has led us to think, well, we might still use transgenes in mouse, but let's try to make everything from naive stem cells and not try to get placenta-derived cells and so forth. Again, it's more complicated. And also thinking ahead for trying to do human embryo modeling, we don't want to use embryo-derived materials, and actually there's no really good trophoblast stem cell lines and so forth. And this is the work really of Shadi Tarazi and Alejandro Aguilera together with Karim Gibran. And their ideas was, let's try to work with mouse naive cells, split them into a pool of 3, and then try to nudge with transgenes and other molecules a fraction to become trophoblast, a fraction to become primitive endoderm, a fraction that is untouched, and we put them together and mount them as soon as possible on the device and the conditions which we know support mouse natural embryos. And what they did actually is really optimize conditions, how to push them to become trophoblasts very quickly, overexpress a transgene CDX2 and inhibit hippopatho with LPA, or do GATA4 expression to make PRE, so we can induce these populations and mix them. And then what happens is the following. You start with 25 cells, and they start growing. You think in the beginning this is just a ball of cells. But at day 4, actually, you realize that something is happening, that this is actually a polarized entity. And if you compare it to a natural mouse embryo, you can see the EXE, which is going to form the placenta on one side, and the epiblast on the other side. And as it grows, you can really see for the first time structures that are really resembling the real embryo. So you can see here the primitive streak. You can see you have the amniotic fold, the amniotic cavity in these stem cell-derived developmental models. At day 7, boom, you have a yolk sac growing, and inside of it, until we can reach day 8.5, at about 0.5% efficiency in development of embryo-like entities. And we've characterized these, again, we're using the same device, the same conditions, although the efficiency is low, but since we can grow millions of stem cells, we can really pick the correct structures and make many of these 8.5 developmental models. And if we look at them, and you can stain them at the different stages and compare them to a natural embryo, you can see that they are expressing the right markers in the right place at the right time. But still, embryo models, as you can see here, have developmental defects that prevent them from going further. A very enlarged heart or a neural tube that is still open and overcurved. But still, this is a pattern formation that is driven by the stem cells, and we are not giving pulses of wind and so forth. So the stem cells are propagating this differentiation and make embryo-like models, which are not identical to embryos, but clearly they can teach us about patterns that happen during embryogenesis. And again, if you look at the structures, this is, you can see here, a heart formation or a brain formation. You can see here even the yolk sac, so the first blood stem cells are formed from the yolk sac, and you can see here the vitelline circulation, suggesting that also extra embryonic tissues are being developed in these embryo models. We look at this by single-cell RNA-seq analysis, and this is the natural embryo, and you can see here the different mouse embryo models, they have the same distribution. There are no unidentified cell types, there are no residual pluripotent cells. really, really high self-driven differentiation. While this is very exciting and there are a lot of questions, we in parallel started to look at human embryo models. As I mentioned before, the reason for that is that we don't know a lot about early human development. We need to remember in these first 40 days, most pregnancy failures happen during these times, most abortions happen in these times, most developmental defects happen during times. Their embryo is very sensitive, like CMV infection, alcohol is very sensitive in these periods because this is when the embryos are formed. And just to put everyone on the same page, this is your typical YouTube movie about how human embryos, in this case it's from Imperial College. So the blastocysts are the same, but once implantation happens in the human, it's very different from the mouse. And you can see here, this is a bilaminar disc. So the epiblast in blue stays flat, and the hypoblast is yellow. This is the yolk sac. And you have this amnion cavity, and this is the amnion rooftop. And if you look at this from the top, you can see this is shapes like a disc. That's why it's called the embryonic disc. And around day 14, you can see here in the posterior part, you're going to get something called the primitive streak, which is really the beginning of gastrulation that happens in the human at this stage. If you look at the section of a later stage, again, this is the epiblast and the yolk sac, and now you're going to see these orange cells that emerge. These are extraembryonic mesoderm cells. And these cells are very important. They make the chorionic cavity and so forth. They are not found in the mouse at this stage. And if you ask, why did the screen kind of darkened suddenly and the cells really emerged, it's because nobody knows where these cells come from. So we cannot animate them. And it just shows you exactly why we are researching this, because we don't know enough about the human development. If even at day 14, we have a whole lineage, we have no idea where it's coming from. So when we took this challenge, we started, and the first obstacle happened to be solved a priori by us, which is we have to start with human naive pluripotent cells. And I showed you, we have already made different versions of human naive pluripotent cells. And the next challenge is really how do we induce these cells to the trophoblast, the hypoblast, and the mesoderm, and we can really do it. And in the human, as I said before, we can do it even without transgenes. And we developed protocols how to induce these cell types. So for example, a protocol is called RCL, which is RPMI media with Wind and LIF. And you can see we induce in the same protocol SOX17 yolk sac cells and BST2 positive extraembryonic mesoderm cells, which are mutually exclusive. So we can get these both cell types. And then a lot of optimizations of how many cell types and how many different media and so forth. And what we came up with, which I'll convince you, is a way at about 1% we can make human embryo models, which are still not identical to human embryos, but they have a lot of properties resembling day 14 human embryo. And in these developmental models, if we start staining them, look at these OCT4. This is the epiblast. This is the yolk sac in yellow, and this is the bilaminar disc, the blue and yellow. This is the amniotic cavity, and this is all surrounded by trophoblast. And this is a light sheet microscopy this is a segmentation of an actual staining, and you can see the structure. Here's the epiblast, here's the amnion on top, here's the hypoblast, and this is the yolk sac. This is the actual staining, and you can see the bilaminar disc. And again, through light sheet, we can go up and down through these different staining and look at them. And really, we've characterized these and at different stages, and this is your textbook structures, and we can really see a lot of similarities of these entities that we are developing just from human stem cells in the dish without using any real embryo-derived material or any aborted material and so forth. If we look at our different compartments, this is the yolk sac, and you can see not only the cavity, but look at the yolk sac. You can see here the visceral yolk sac, the epithelium is columnar, versus in the parietal yolk sac, it's actually flat. And if you look in the Carnegie collection, again, we don't use any real embryos in the study. The atlases are good enough, and you can see the structures. And what this shows you that self-organization, that the cells are assuming really fine details of about what is happening in between them. So I want to push this and try to show you that there are other structures, which is, for example, the posterior epiblast that has, you can see the stalk, which is a structure in the human, and you can see brachyury in the posterior part, which is really show that these embryo models about to gastrulate and develop into different structures. And in the last slide that I want to show you is really that you can see that the placental cells are also developing other structures. And you can see here, these are going to be called lacunas. So you're going to see now in this video, this is the placenta. And these are the lacunas. So they are interstitial kind of intercellular spaces that typically get filled with blood. And again, this is not in the mouse. And this shows you how these stem cells can assume properties. And we can start developmental questions about how the placenta is formed at these early stages of development, which is our main goal. With that, I've finished my talk. And unfortunately, I haven't answered any of the questions I started with about naïve and the prime. But this is, as Pavel mentioned, we are basic science. Of course, we will get back to that, and we are driven by our curiosity. But we're very excited about this type of research because it really allows us in a more ethical, approachable, realistic way to look at development from different species, including the mouse, where our knowledge is limited. And this type of research really involves, it's going to require a lot of scientists. Already great colleagues are working on this. I've made also great contributions, and I'm definitely standing on shoulders of giants that have made a lot of this knowledge available. And really, we hope in the next 10 years that we can solve issues such as the low efficiency of this process and so forth, so that really we can make this the entry into this field and make this a very widespread tool to look at development in a new way. Thank you very much, and happy to take questions.